Hello, my name is Timothy Clark, and I will be doing a brief overview on the Nipah virus as well as outlining possible steps for the outbreak investigation of the virus. The Nipah virus was first documented in 1998 in a town in Malaysia called Kampung Sungai Nipah. This is where the virus got its name. The first outbreak in Malaysia mainly consisted of pig farmers, which led to the hypothesis and observation that an intermediate host of the Nipah virus was pigs. After further investigation and research, fruit bats were also established as being hosts and reservoirs for the Nipah virus. From 1998 to 2004, there were reports of deaths related to or caused by the Nipah virus that spread from Malaysia to Singapore and Bangladesh. As reported by the World Health Organization, the Nipah virus has not been reported outside the Southeast Asia region. The Nipah virus is categorized as a zoontic paramyxovirus. Nipah virus leads to a condition known as severe encephalitis, which is inflammation of the brain. Symptoms and signs of encephalitis include confusion, agitation, or hallucinations, seizures, loss of sensation or paralysis in areas of the face or body, muscle weakness, problems with speech or hearing, and loss of consciousness. The main mode of transmission has been researched to be direct contact with infected tissues and fluids. The risk factors associated with Nipah virus mainly include direct contact with infected individuals or animals that have relation to the Nipah virus and antibodies. This graph shows associations with infected individuals and animals. The geographic distribution of the virus from 1998 to 2017 has been fairly isolated. As stated before, the virus has not left the Southeast Asia region. This map shows the distribution of the virus over the region and concurrent time period associated with the outbreak. The prevalence of the Nipah virus has been isolated to Malaysia, Singapore, India, and most significantly Bangladesh. The World Health Organization report shows prevalence of positive cases reported in each region. Similarly, this report shows the fatality rate of the disease in each region. In total, there were 280 positive cases in the region, 211 deaths, and a fatality rate of 75%. To oversee, prepare, and plan the outbreak investigation for Nipah virus, we will first establish a Nipah virus Control and Prevention Committee, or NVCPC. Due to the geographic isolation of the disease, to this point, the committee will primarily focus its effort in Southeast Asia. One of the first steps for the NVCPC will be to review the laboratory and antibody analysis in the region for the cases that were reported positive. They will do this to evaluate the validity of the new disease. The second thing the committee will do is plan a course of action to establish prevalence data in the region from 2015 to 2017. They will use resources like lab and medical records, as well as a boots-on-the-ground campaign. The NVCPC will then use the collected data to establish the prevalence of the disease during this time period. To address future cases and to better establish preventative methods, the committee will work to establish geographic and demographic information at-risk areas by conducting a questionnaire throughout the region. Using the data received from the questionnaire, the committee will establish at-risk populations and potentially exposed targets. They will run lab tests to analyze the extent of the exposure in these target populations. The last focus of the NVCPC will be to evaluate the environmental factors in the region that could host or be a reservoir for possible Nipah contamination. Areas that will be assessed are farms, livestock and feeds, and the local ecosystems, particularly the fruit bat habitats. Thank you for your time.